This procedure begins with the staging of zebrafish embryos. When the embryos have reached the stage of interest, remove them from the chorion. Next, holes are created in an agarose-coated dish with a pipette tip for mounting the embryos, and the embryos are placed gently in the holes with their tails down. After the embryos are mounted, a fluorescent dye is carefully injected into the ventricle space of the embryo through the hindbrain. After ventricle injection, the embryos are imaged and the data is processed using Photoshop.
Hi, I'm Jennifer Gutzman from the laboratory of Dr. Hazel Siv at the Whitehead Institute for Biomedical Research and MIT in Cambridge, Massachusetts. Today we will show you a procedure for zebrafish brain ventricle injection. We use this procedure in our laboratory to study brain ventricle formation and neuroepithelial morphogenesis in live zebrafish. Due to the nature of transparent zebrafish embryos, characterizing the brain ventricle shape can be difficult. This technique is a useful and simple way to differentiate the fluid-filled space from the surrounding tissue and to characterize mutant embryos that may have brain ventricle formation defects. So let's get started.
To prepare for injecting the zebrafish brain ventricle, we start by making the injection needles. A Sutter Instruments needle puller is used to pull the capillary needles. The capillary needle is then filled with a fluorescent dye, such as Texas red dextrin. Next, the filled needle is mounted in a micromanipulator and microinjection apparatus. Cut the needle to the appropriate size at an angle to create a beveled tip. Measure the drop size of the needle and check that it is between 1 to 2 nanoliters per injection in oil. To begin this procedure, stage the embryos according to Kimmel et al. Embryos should be injected 30 minutes earlier than the stage of interest. For example, to study the ventricles at 24 hours post-fertilization, inject the embryos at 23.5 hours post-fertilization. The embryo at the stage of interest is placed under the stereo microscope and the chorion is carefully removed from the embryo using forceps. Next, an agarose dish for the embryos is made. Using a plastic dish coated with 1% agarose, poke holes in the agarose with a plastic 1 to 200 microliter pipette tip. Carefully remove the agarose plugs from the holes using forceps. Transfer the embryo and embryo media into the agarose dish with holes. Add tricaine to the media to anesthetize the embryo and prevent movement during the experiment. Gently place the embryo tail down into the hole and orient it so the injection apparatus is on the posterior side of the embryo. Now that the embryo is placed and oriented correctly in the agarose dish, we are ready for the brain ventricle injection. First, arrange the micromanipulation setup so that the needle tip is in the same field of view as the embryo on high power. Carefully put the needle through the thin roof plate of the hindbrain just posterior to the R0-R1 hinge point without hitting the brain tissue below. Inject just enough dye to completely fill the ventricles. It may take several injections to fill the ventricle space depending on the stage of the embryo. When the embryo has been injected and is ready for imaging, a new agarose dish for the embryo is prepared. Using a clean 1% agarose-coated dish, poke holes in the dish and remove the plugs. Transfer the embryo to the dish and add tricaine to anesthetize the embryo and prevent movement. Place the tail of the ventricle-injected embryo into the hole in position for a dorsal image. The embryo is now ready for imaging. Take an image using transmitted light. It is very critical to not move the embryo or the microscope. Next, change the settings on the microscope and take an image with fluorescent light. Reposition the embryo to take lateral images with both transmitted light and fluorescent light. Save all the images as TIFF files for image processing in Photoshop. To process the images in Photoshop, open the transmitted light and fluorescent light images of the same embryo taken in the same position. Be sure all of the settings are the same for each image. Drag your fluorescent image onto the transmitted light image and line them up exactly. With the fluorescent image selected, select Image, Adjustments, and then Replace Color and change the black background to white. In the Replace Color window, adjust the fuzziness factor to the right until the fluorescent image looks uniform in color. In the Layers window, select Multiply to view the overlay images. Your ventricle space image should match the transmitted light image exactly. Save this file and repeat for any other images. If the ventricle injection is performed correctly, the images should have sharp edges and non-diffuse dye, as shown here in these images of 25 hours post-fertilization wild-type embryos. Panel A shows the bright field image, Panel B shows the fluorescent image, and Panel C shows the overlaid image. A lateral view of the overlaid image is shown in Panel D. On the other hand, if during the ventricle injection the needle is inserted too far, it will enter the brain tissue below the ventricle space and lead to an incorrect injection. This results in dye that is visible outside of the ventricle space and in the yolk, as shown here. Panel E shows the bright field image, Panel F shows the fluorescent image, and Panel G shows the overlaid image. A lateral view of the overlaid image is shown in Panel H. The arrows indicate regions where the dye has gone beyond the brain-ventricle space.
In this video, we demonstrated how to inject fluorescent dye into developing zebrafish brain ventricles. This method is used to visualize the brain-ventricle space in contrast to the surrounding neuroepithelium and is extremely useful for determining the shape of the ventricle space as well as the shape of the surrounding brain tissue. This technique allows us to better understand the process of brain-ventricle formation and brain morphogenesis over time in the live embryo. It is an excellent tool for studying brain-ventricle formation defects and for initial characterization of brain morphogenesis mutants. So that's it. Thanks for watching and good luck with your experiments.